Okay. Okay, how many of them are there? 3 of you are there. So we have to complete the chapter on seeds, no? Seeds is left. Till which part of the chapter we are done? I think we are done with the differences. Albuminous, ex-albuminous seed, the structure of the bean seed, the structure of the maize seed. No, that much we are done.
Yes.
Yes or no? Now we are left with only the structure of the seed. The structure of the seed. No, we are left with the process of germination.
We have to start the experiments.
No, process of germination we are not done. Process of germination मैंने नहीं कराया.
On the condition that was discussed.
The last class we had only finished. We had already finished with the structure of this bean seed and the maize seed and their differences. All these we finished, no? And I said the 2 topics which are left are the experiments and the process.
Yes.
Epigel, hypogeal germination process is left, and what we are left with only the experiments. So that part is left now.
Yes.
First of all, we'll look into the experiments. Okay, first of all, we'll who is sharing?
This. Yes.
Okay, I'm not able to share anything here. Okay. 8 minutes.
Because this is
But then even if I am in the classroom, I'm able to share. Acha, anyways, now look into the experiments. Experiments are very easy. Even if I don't show you the pictures, you are able to understand. I was going to show you the images. Now look into the steps of, look into the images, or look into the experiments that are there in your, look into the, I'll get the textbook. Wait, I'll tell you the experiment. The first experiment that we are going to discuss would be germination of seed. Those 4 processes we are going to discuss first. The method of germination, the processes of germination. So let us complete with those experiments. So what is the first experiment about? The first experiment is about, taking seeds soaked in water. So that's the first experiment. So we are talking about, look into the first experiment. Can you see the first experiment about germination is placing the seeds in. Wait, let me just check and tell you. Yeah. So we are now talking about germination experiments. The first experiment I talk about, the first experiment, germination, where we are supposed to understand that water is necessary for the process of germination. Now, can you see? Look into that page of the textbook. The first experiment, is it 8.4 in your book also? 8.4 sub number 1 experiment, that is? Yes, is it that? You have the textbook with you? Anybody is having the textbook with you?
Yes.
Okay, okay. Now, if you have the textbook with you, now let us talk about the first experiment. I'm not able to share the images, so you refer to the images of the experimental setups in your textbooks or the volume. Okay, of the textbooks or the volume. The first experiment that I discussed would be water is necessary for germination. I'm sorry. Water is necessary for germination. Can you see that? Water is necessary for germination. Now, water is necessary for germination. What is the experiment about? I talk about in 2 different conical flasks, we are taking seeds in 2 different manner. So in the first conical flask, what are we taking? In the first conical flask, we are taking seeds which are soaked in water. In the first conical flask, we are taking seeds which are soaked in water. And in the second conical flask, what are we taking? Seeds which are dried seeds. So now, what are we doing? What is the setup? We take a conical flask. We take wet cotton wool. What is the meaning of wet cotton wool? It's a bed of cotton which is kept moist. A bed of cotton which is kept moist. And above that, some soaked seeds kept. What about the second conical flask? In the second conical flask, we take the dry cotton. We don't wet it. We don't keep it moistened. We just take the dry cotton bed. And above the dry cotton bed, what are we doing? Keeping some seeds which are not soaked in water. Not soaked. Okay. And then we wait for some hours. After some hours mean, remember, it has to be a minimum of 8 to 10 hours. In a period of some 8 to 10 hours, what my understanding is that the seeds which are kept in, the wet cotton wool, seeds which are kept in the wet cotton wool undergoes the process of germination. The seeds which are kept in the wet cotton wool, they start undergoing the process of germination. They start undergoing the process of growth of the shoot, the radicle, the plumule. But what about the second conical flask? What do you think? Should they also show the method of germination or not? No, even if you're waiting for 8 to 10 hours in the second conical flask, we get to see that in the second conical flask, we get to see that nothing is coming on. No process of germination is happening. No process of growth of the shoot is happening at all. No process of growth of the shoot is happening at all. And now from this, what are we understanding? That for the process of germination, that wet cotton was playing a major role, no? Because in both the conical flasks, the difference is in one we have kept the wet cotton wool and in the other we have kept the dry cotton wool. Then what is the explanation that you are going to write to justify that in the wet cotton wool, the conical flask with the wet cotton wool, the process of germination happened? What are you going to write? The clarification is, the justification is that yes, water is necessary for the process of germination. Water is necessary for the process of germination. Water is necessary for the process of germination. So yes, we now understand from the first experiment that water is very much necessary for the process of germination. So that's the reason whenever we keep seeds in the dry environment, they don't undergo the process of growth. But when we are keeping them in a wet, moist environment, they start the process of growing into a shoot. Come to the next experiment. Now that we are talking about, again, I take a conical flask, or I take 2 parallel conical flasks. So in these 2 conical flasks, I'm going to keep seeds in 2 different manner. In one setup, I'm going to take, in one conical flask, I'm going to take some soaked seeds and place that conical flask in the refrigerator. Place the conical flask in the refrigerator. The second conical flask, what are we doing? The second conical flask, I've taken soaked seeds, but I'm just keeping it at room temperature. I'm not keeping it in the fridge. I'm not keeping it inside the fridge at all. So what do you think? The conical flask 1, where I have kept it inside the fridge, or the conical flask 2, which is actually kept outside at the room temperature, which one should allow? Ashita and Bhakti, something is fishy there. Yes, Ashita, your concentration seems to be diverted. Would you prefer to go to the next bench?
Which one?
Ashita, would you prefer to go to the next bench and sit? Because as it is, since I'm online, and on top of that, if you both are getting distracted, it's difficult. So now what I said in the second setup, what is the second setup? In the second setup, what I was saying, the conical flask with the soak seeds, did we keep it in the refrigerator? No, we had kept it at room temperature, and that's the reason we saw that the process of germination happened. So ultimately, what is the difference? The difference lies here, that in the first conical flask, why did the seeds not germinate? Because the temperature in the fridge is always lesser than 10 degrees. And the optimum temperature which is necessary for the process of germination is always something around 20 to 30 degrees. So optimum temperature which is necessary for the process of germination may be something around 20 to 30, 35. So that is the optimum temperature for germination. And now that we are talking about that, as a result of the temperature in the fridge, which is so low, much lesser than the optimum temperature, the enzymes which are necessary for the process of germination remained inactive. The enzymes remained inactive, and that's the reason the process of germination could not happen. Third concept is, what type of experiments can help us to understand that oxygen is very much necessary for the process of germination? Which type of experiments? I talk about, again, two conical flasks. Could you see those figures that I'm talking about? Now this, you have a figure there in your book.
Yes, ma'am.
Saw that figure? Yes. Now for this experiment, you have got a figure in your textbook. Which experiment I'm talking about? Oxygen is necessary for the process of germination. Now what is happening there? Again, we take two conical flasks. Every time we are taking two conical flasks. So again, we take two conical flasks. Again, we take two conical flasks, and in one, we take some soaked seeds and the wet cotton wool. And in the other also, we are taking the wet cotton wool and the soaked seeds. So it is expected that yes, both the type of seeds, both the seeds in both the conical flask, should undergo the process of germination. Is not so. So that is expected. But in one test tube, in one conical flask, what is inserted? A test tube containing water. And in the other conical flask, meaning if I talk about conical flask B now, conical flask B, inside conical flask B, inside conical flask B, a test tube is inserted, and that test tube contains alkaline pyrogallic acid. What does it contain? Alkaline pyrogallic acid. Alkaline pyrogallic acid. So now this alkaline pyrogallic acid, which has been used in this experiment, is generally known to absorb oxygen in whichever environment it is placed. Alkaline pyrogallic acid is known to absorb oxygen in whichever environment it is kept. So now, in the first conical flask, we took what? Soaked seeds we took. And what was there inside the conical flask? What was there inside the conical flask in the first setup? In the first setup, there was water. In the second setup, there was pyrogallic acid. No, in the second setup, there was pyrogallic acid. And I also mentioned just now that pyrogallic acid can start absorbing oxygen and not allow the seeds to germinate. So if these theories are correct, then we should understand that the second test tube, which has the pyrogallic acid and it is kept in close contact with the seeds, then obviously the oxygen of the inner environment is all trapped by the pyrogallic acid. The oxygen is all trapped by pyrogallic acid, and are the seeds getting anything? No, the seeds are hardly getting any oxygen. So the process of germination is not happening there. Not happening there. Next is you'll talk about the 3-bean experiment. Did you see that 3-bean experiment? Did you see that 3-bean experiment?
Yes.
Now, in the 3-bean experiment, what are you supposed to write down? In the 3-bean experiment, you come across a very simple experimental setup where we take a beaker almost 3/4 filled with water. We take a beaker almost 3/4 filled with water, and then next one, take a plank or a wooden strip and attach 3 seeds in such a way that one seed should be completely above the water. The second seed should be completely drowned in the water. And the third seed should be now, the third seed should be just placed at the junction of the water, at the junction of the air and the water rather. So just at the level of the water, the third seed has been placed. So if that is so, then which seed is supposed to show the process of germination? Which seed is supposed to show the process of germination? Can the first seed at the bottom, completely submerged by the water, can the first seed show the process of germination? Why not? What is lacking there? Why the first seed, which is completely drowned in the water, why can't it survive? It can't survive, it can undergo the process of cell division because the seed at the last, the seed at the bottom, it is getting enough water. The seed at the bottom is getting enough water. The seed at the bottom, is it getting air, oxygen? Oxygen, is it getting adequate? The seed at the bottom, is it getting adequate oxygen? No, it is not getting adequate oxygen. No, it is not getting adequate oxygen. But water it is getting, temperature is ambient, so it should have undergone the process of germination, but it does not. Then look into the third seed, which is completely stuck into the plank only. Completely stuck into the plant only. Now, what is the meaning of this? The meaning of this is, now, whenever we are talking about that the seed exactly at the top, what are the factors it is getting and what are the factors it doesn't get? Can you tell me? The seed at the top, the third seed, or if I place the other way, first seed, or the seed which is completely out of touch of the water, which are the factors it is getting? It gets what? Sunlight, does it get? Yes. Temperature, does it get temperature? Adequate amount of temperature, does it get? Yes. Then adequate amount of water, does it get? No. Adequate amount of water or moisture it is not getting. Adequate amount of water, moisture it is not getting. So since it is not getting adequate amount of water, moisture, the process of germination, that's why it's not happening. But what about the middle one, the second seed, the middle seed? Now the second seed, whether you talk about this particular seed directly in contact with water, a little bit in contact with water, or at least considerably in connection with the water, in any of these cases, it will start the process of germination because the middle seed is getting adequate amount of the temperature. It is getting adequate amount of sunlight, oxygen, and on top of that, just the right amount of water that is necessary for the process of germination. So this 3-bean experiment is very common and you should always remember to study this well. Then the text exercise, did you do? I'm going to take the last topic that is Epigel Hypogeal that we have to study. But before that, I want to complete with few more questions from this part. Did you do the progress checks, etc. in the textbook?
Yes. Yes.
And Okay, so if you have done that, progress check. Now, look into your textbook. Look into your textbook. The multiple choice question, the first question. What is the first question? Read out, multiple choice.
Which one of the following plant part is correctly matched with one of its stated characteristics? A.
Ah.
Dash alluvial layer. B. Endosperm. C. Maize Grain. Colloidal. D. Wheat grain exhalation.
Okay. Try this question. Try this. You have to understand that with one of the above characteristic, which causes the following plant factor is wrongly matched. Wrongly matched. Which one is wrongly matched?
No, ma'am. Three are wrongly matched, only one is correctly matched. You have to find which is correctly matched.
Which is a correctly matched? Mango seed and elurone. Is it correct?
No, ma'am.
No. Number B. Bean seed and endosperm.
No, ma'am, not correct.
No. Maize seed and cotyledon.
No, ma'am. Maize seed and colloidal.
Correct. And wheat grain and
Exhalation.
okay. So see that all of you get this answer. Next is try name the following. Try this for name the following. Name the following. What is the first one? Cannot exert enough force to push the soil upward. Nay. This is not, I think, a monocotyledonous endospermic seed. What is the answer? Maize. A chemical used in experiment which absorbs oxygen. Pyrogallic acid. Okay. Part of the plumule above the embryonic axis. Epicotyle. Done. Fill in the blanks, Carlo. All these parts you have finished already. Carlo, some exercises. Fill in the blanks.
What is the name the following F1? A seed with folded finger leaves.
What is it? Which number?
Name the following F.
Name the following. But which question you are talking about? My question numbers would be different. Mine is an old book. Tell me. I did not hear you correct. I did not hear you clearly.
Seed with folded plumule leaves.
Acha. Seeds with folded plumule leaves. Seeds with folded plumule leaves. And the question is, are you supposed to ask, are you supposed to write down the type of seed with folded plumule leaves?
Yes, name the following. Now it will be bean seed, now.
That will be folded plumule. Folded plumule should be the no maize. Folded plumule is maize.
Oh.
Oh no, folded plumule is bean. No, dicot. Folded plumule is bean. Dicot, correct. Folded plumule would be bean, except for the dicot it is. Okay, so this part is done. The experiments we are done. Now, next is we move on to the method of the types of germination. Let us move into that topic. Types of germination. So here we are going to learn two major methods of germination. What are the two major methods of germination that we're going to study? The type of germination, the methods of germination are called epigel and hypogeal. Method of germination would be called epigel and hypogeal germination. Now, in each of these methods, we are going to come across that either now you are already, I have already explained you in the class what is meant by the hypogeal and what is meant by the epigel. Epigel is a type of it is actually the extension of the radicle. It is, first of all, we talk about that the small portion which comes up, it takes an arc and then it again takes up the seed upwards. So for this, first of all, you'll keep the diagram in front of you and you'll take out your notebook. You take out your notebook. I will first of all give you the entire method that step by step you should write. And then you are going to match it up with the diagram, because I'm not able to explain it with the diagram. Okay, write down the head. Write down the head. The first method of germination that we're going to study would be hypogeal germination. We start with hypogeal. Write down hypogeal germination. Hypogeal germination. Under hypogeal germination, you write, or within bracket, you may write pea seed. Hypogeal germination of the pea seed we are learning first. Hypogeal germination of pea seed. Point number one, the seed absorbs water . The seed absorbs water and swells up. The seed absorbs water and swells up. Point number two, seeds in the water swells up. Number two, food reserves in the cotyledon. Food reserves in the cotyledon become soluble by the action of enzymes. Food reserves in the cotyledon become soluble by the action of enzymes and pass into the tips of the growing plumule, and radicle. Growing plumule and radicle. Next point. This growth begins to elongate Okay, did you write down the first point? I'll repeat again. Food reserves in the cotyledon become soluble by the action of the enzymes and pass in to the tips of the growing plumule and radicle. Finished till here?
Yes.
Third point. So these are the steps and the entire process of epidural and hypogeal, which is not clearly that much given in your book, in this book. So that's why I'm giving you from another book there, so that in your exam you can easily write. Number 3, the epicotyle belongs to elongate. The epicotyle belongs to elongate and form. Epicotyle belongs to elongate and form loops, L-O-O-P-S, loops, and form loops. Full stop. Root hairs appear in the region of the radicle. Root hairs appear in the region of the radicle where elongation the region of the radicle where elongation has ceased. Where elongation has ceased. Ceased means to stop. Where elongation has ceased. I'll explain you with the diagram. You first write down the steps. Epicotyle elongates radically. Epicotyle elongates radically.
Next point.
Next point, correct. Next point. Epicotyle elongates radically. Epicotyle elongates radically, comma, the plumule located at its tip. Plumule located at its tip. Plumule located at its tip. Plumule located at its tip become free from cotyledons. And straighten to move upwards. Straighten to move upwards. Next point. Young leaves appear. Young leaves appear and prepare food. Young leaves appear and prepare food. Full stop. Next point, seed coat and cotyledons. Seed coat and cotyledons remain in the soil. Seed coat and cotyledons remain in the soil. Last point, primary roots persist. Primary roots persist giving rise to taproot system. Giving rise to taproot system. Now, look at the figure to understand what you wrote. The diagram of germination of the pea. Now I'm considering germination of the pea, and the germination of the pea is what type? Germination of the pea is what will you say that is? What type of germination? Hypogeal. Hypogeal germination. So follow the figure now. The seed is in the soil. The seed is getting adequate amount of moisture. The seed is getting adequate amount of water. As the seed gets adequate amount of moisture, the cells of the seed swell up and they start bursting the seed coat. But now that the seed coat has burst out, what happens? Which tissue first comes out? It is the small radicle which will come out. It will be the small radicle which is coming out. But on the other way, it is also seen that the plumule starts coming out. It is also seen that the plumule starts coming out, and it is the epicotyle which is elongating much faster. And this epicotyle which is elongating much faster, how does it behave now? This epicotyle is starting to move above the soil. The epicotyle, which is now growing much faster, starts growing. But by the time is the radicle elaborately functional? Just check your diagram. Keep checking your diagram. Keep checking your diagram. Not able to share, otherwise I wanted to show this diagram. You have no that diagram of germination of the pea in your book. You have no that diagram. You have all three of you have that diagram in front of you, no? So take the diagram. So I'm explaining based on that diagram that if I talk about the PC, the method of germination is hypogeal. So what are the steps of hypogeal germination? The seed which is in the water, which is in the soil will soak up water, and the seed coat will rupture. As the seed coat ruptures, then what happens? The radicle first comes out and starts little bit forming some fibrous roots. Then by the time next, what happens? The epicotyle starts elongating. The epicotyle, where is the epicotyle? Above the embryonic axis, no? The upper part of the embryo, epicotyle. Now the epicotyle starts coming out, and later this epicotyle, which is now coming out, and slowly it starts giving rise to the shoot, that is mainly the stem and the fruit. Then this epicotyle, which is elongating slowly, it has also the plumule in it. It also has got the plumule in it, and this plumule, it also has got the plumule in it. And now next what? This plumule starts growing, and as this plumule starts growing, ultimately we get to see that an arc-like structure is formed, and very fast now the new leaf starts coming up. That is the entire epicotyle has started giving rise to the shoot. But later what happened at the region below the soil? At the region below the soil, the radicle started growing, and the radicle started giving rise to the root system, and the hypocotyle, which was below the embryonic region, that has started giving rise to the entire root system there. So what are the prominent things you come across in this method of germination? Did the seed actually come above the soil? Did it come up above the soil in this method? No, the seed remains below the soil. The seed remains below the soil. Now, is this method of germination carried out in dicot seeds or monocot seeds? Is it basically carried out in dicot or monocot? When we are talking about this method, hypogeal and dicot seed is the most common concept of germination. So follow the diagram. What is happening? Only that concept of hypocotyle and epicotyle you will find little bit disturbing, confusing. Otherwise, the features straight away are the seed will never come out of the soil. The seed is always inside the soil, which will later perish away and provide nourishment. So if in a method of germination, the hypogel elongates faster, that is what is called, the hypocotyle elongates faster, that is what is called the hypogeal germination. Could you understand this? Check the figure, what are the features and what is the sequence? Now, next paragraph you write, Epidual germination. I'll give you the example of a pea. Epidual germination in pea. What are the steps? Epidual germination in pea. Epidual germination. Now, for epidual germination, you take out the diagram of the bean seed. So far, we were talking about the pea seed. Now, you take out the diagram of the bean seed. Take out the diagram of the bean seed now. Refer to that diagram. Take out the diagram of the bean seed and we'll study from the bean seed now. Now, in case of the process of epigeal germination of the bean seed, wait. Now, in this case, a minute. Hypogeal germination in the pea we have done. So in the maize, we are not repeating, it's almost the same thing. We had been talking about hypogeal. Now, next is we have to talk about epigeal method of germination. Write down. Maize. A minute. Just now we completed about the hypogeal germination in the pea. Now I give you about the epigeal germination. Just give me a minute, 1 minute. It's lost. Hypogeal is this one. Epigeal in bean, we are supposed to study. Wait a minute. Write down. The stages of epigeal germination. Write down the epigeal germination. Put the next heading. Epigeal germination in bean. We did hypogeal germination in pea. Now epigeal germination in bean. Epigeal germination in bean. Now write down the stages of epigeal germination. The stages of epigeal germination are, number one, stages of epigeal germination are, number one, seed absorbs large quantities of water. Seed absorbs large quantities of water. Seed absorbs large quantities of water and swells up. Seeds absorb large quantities of water and swell up. Number 2, the seed coat softens. The seed coat softens. Seed coat softens and ruptures. Seed coat softens and ruptures. RUPTURES. Softens and ruptures. Ruptures. Number 3, the radical pushes. Radical pushes through the seed coat. Radical pushes through the seed coat, bends downwards and grow into the soil. Bends downwards and grow into the soil. Bends downwards and grow into the soil. Third point, radical forms primary root. Radical forms primary root and lateral roots and lateral roots begin to emerge. Full stop. Root hairs appear. Full stop. Next point. The cotyledons are within the ruptured seed coat. Cotyledons are within the ruptured seed coat. Within the ruptured seed coat. Next point. Hypocotyle elongates. Hypocotyle elongates. Hypocotyle elongates, pulling the cotyledons up into the air. Hypocotyle elongates, pulling the cotyledons up into the air. Pulling the cotyledons up into the air. Next, hypocotyle becomes completely, the hypocotyle becomes completely straight. Hypocotyle becomes completely straight and possesses plumule at the tip. Possesses plumule at the tip. Next is the young leaves. Young leaves folded at the tip. Young leaves folded at the tip. Start unfolding. Start unfolding. And shoot is developed. And shoot is developed. And shoot is developed. Okay. So now look into the diagram as I explain you. Look into the diagram, all of you. Try to understand. The first one did we study? Hypogeal germination. Here what is happening? First of all, we get to see a small amount of root hairs coming out. These are called, this small root hairs would be called the radicle. It's a part of the radicle. Then next what happens? Follow the diagram. Next what is happening? The radicle is growing little bit. And again, the radicle is growing little bit and it starts now moving into the soil. Next, what happens if you now look into the pea seed I'm talking about? Next, what happens? Next is some dividing tissue starts rupturing from the wall of the seed coat, and when you are talking about this, okay, so I told that the radicle is going down. Now look into the upper part, the third figure I'm talking about. Could you see the word testa there? The testa breaks up, and now what will happen? The epicotyle will start emerging, and it starts moving above the soil. As it starts moving above the soil, it breaks up the testa, breaks up the seed coat, and starts moving above. But as it starts moving above, it more or less, first of all, takes a arc-shaped structure. It more or less takes a curved structure, arc-shaped structure. So what I was saying, where's the Shreya gone? Why were you going? Why were you going out? Acha. So now ultimately what happens? Now look into the second diagram. The second stage is the epicotyle takes an arc, the epicotyle becomes bent, and it starts moving above the soil, and it takes an arc-shaped structure, almost holding the soil. But what about the seed? Is the seed also coming along? No, the seed is inside. The seed does not come along. Then this epicotyle keeps on elongating, and the epicotyle has which tissue at the tip? The epicotyle has the plumule at the tip. And now it keeps on going, and it starts elongating all throughout, resulting in the formation of some leaves, folded leaves first, and then some leaves. But what about this cotyledon still? Is the cotyledon above the soil? No, the cotyledon is still inside the soil.
Above the soil.
What are some differences you try to come across? What are some differences in the epigeal germination? In the epigeal germination, when we are talking about it, as the radicle develops, but immediately, are we talking about the radicle and the hypocotyle developing from two different ends? No, it is the after the radicle emerging out, it is the hypocotyle again which also starts emerging out. Then the hypocotyle starts elongating in such a way that the hypocotyle becomes extensively straighten up. The hypocotyle grows extensive. And now look at that figure. The bean seed I'm talking about, the hypocotyle starts growing extensively. And as the hypocotyle starts growing extensively, also resulting in the form of an arc-shaped structure, it carries the seed along with it out of the soil. It carries the seed. So it carries the seed out along with it. And now the seed is no more, the cotyledons are no more in the soil, rather the cotyledons are all suspended in the air. But immediately, in short period of time, what happens? The two cotyledons, they would just emerge, they would just open up, and leaf-like structures would emerge out. They would just open up and leaf-like structures would emerge out, okay? They would open up and leaf-like structures would emerge out. And slowly, what happens from these leaf-like structures, which are actually the first set of leaves, from this region axis, the rest of the shoot will all develop. So that is about the shoot part. What about the cotyledons which were coming above the soil? What will happen to these? these cotyledons actually appear as green leaves for earlier. These cotyledons appear as green leaves earlier, but within a short period of time, these cotyledons become brown, they die out, they become papery. So after some time, are we able to see these cotyledons anymore? No, we are not able to see these cotyledons. These are already died out and shriveled off. These are gone. And now the root system, the roots will start again flourishing in the soil. So let us now write down few differences. Now take in your book also, there is a table given. Could you see epigeal and hypogeal germination? What are some of the major points to remember? If I talk about the epigeal and the hypogeal germination, look into the first point. The most important point between these two is about the structure of the cotyledon. Where is the cotyledon? The cotyledon is inside the soil. In which type? Epigeal or hypogeal? The soil contains the cotyledons. In which one? Epigeal or hypogeal? It is in the hypogeal. Epigeal cotyledons are coming above. And what is the second difference you can say? If I talk about just now that I was talking about the hypogeal germination, what did we come across? Yes, in case of the hypogeal germination, the epicotyle was elongating rapidly. And now that I talk about the bean seed, the hypocotyle was elongating rapidly. It's not so. The hypocotyle was elongating rapidly. So this is about the basic concept of epigeal and hypogeal. With the diagrams, you are supposed to understand. And diagram identification also you should understand. You will get this diagram to identify which type of germination it is. What is the simplest way to remember which type of germination it is? If you get to see the seed inside the soil, it is always hypogeal. Seed away outside the soil, epigeal. Let us talk about the last topic there, vivipary or viviparous germination. What is vivipary or viviparous germination? When I talk about vivipary, it's a method of growth of the plants. In this method, what's happening? New buds develop, the process of growth also happens. No fertilization, but without fertilization, the plant parts are also giving rise to new plant parts. But the entire process of growth of the new plant, is it happening after detaching from the parent body? No, the entire process of growth of the plant parts continues and happens and takes place, remaining in contact with the plant body, remaining in contact with the parent body. So this new plant is formed, remaining in contact with the parent body. The young plant remains in contact with the parent body, and such type of method of germination is known as vivipary, and the plants are known as viviparous. Plants are known as viviparous. Examples are what? All the different type of marshy plants, Soneraceae, etc., etc. All these different type of Rhizophora, all these different type of plants. These are. Now you have got some diagram-based questions which you are going to do. Which are the diagram-based questions in your book? Look at the last section of the chapter, the question section of the chapter. Look in the question section of the chapter. First of all, after you look into this, fill in the blanks. Did we do the fill in the blanks? No, we haven't done. Fill in the blanks pehle karenge. Complete that. Fill in the blanks, and some diagram-based questions I'm going to give you. Some diagram-based questions I'm going to give you. What is it? Bit louder.
Ma'am, in the volume, the maize grain picture study there.
Maize grain, when you talk about the maize grain, it is the same concept of hypogeal germination. No, maize grain also monocot it is, but though it is monocot, we are only to understand one concept, the same thing only, whether we are talking about maize or we had been talking about pea. So difference yehi hai, pea is dicot and maize is monocot, but the method of germination is same, hypogeal germination. So if it is hypogeal germination, what did you come across? The seed or the cotyledon is inside the soil. The method exactly the same. Okay, complete karo that, fill in the blanks. And now the questions. What are the next set of questions in your book? Fill in the blanks is one that I can see. Diagrams hai, diagram-based questions hai.
In the volume.
In the textbook. Achha, you all don't have, all of you don't have the textbook.
We have in the textbook multiple choice. very short answer type. उसमें state true or false, read the following, fill in the blanks, arrange the following sets in order.
अच्छा, fill in the blanks complete करो, fill in the blanks करो.
I mean, short answer type and long answer type. अब question study start हो.
अच्छा, ठीक है, fill in the blanks पहले complete करो. Then long answer type would be the same descriptive process and experiments only. I want to share with you this experiment based questions. Let me see if I'm able to do. Karo. Complete. Let me check.
Our screen is sharing, ma'am, so you won't be able to share. And for that, you have to stop sharing.
Acha, I'll give you some, once you are done with this few questions of your book, you tell me. Then in that case, I'll have to dictate only some questions. I'm not able to share anything. I'm not able.
We have to stop sharing from here.
So karo na. Karo, stop share karo.
Not able to see us.
Then you will be doing so many mischiefs. I'll not be able to see you. That's also one thing.
No, ma'am, I can turn that computer screen pointers.
Karo, if you can do karo, then I can show you those questions. You can solve those questions, no?
I mean, नहीं होगा और नहीं होता.
हाँ, ठीक है. ठीक है. By the time I show you this, you do your this one. Complete your text questions. Are you able to see anything? No. क्यों नहीं हो रहा?
Ma'am, can you give us a 5 minutes break?
Take. By the time I'll check if I'm able to do this. Are you able to see now?
Yes, ma'am.
Now you're able to see. Chalo, after the break, Patafat will do some few questions and then we are done with this chapter. All of you are back. All of you are back. Are you all there now?
Yes, ma'am, we are there.
Shreya, Bhakti, and Chalo. Start solving number 12, number 13. You can see, no? Number 13 solve karo. Number 13 karo. Solve number 13. Can you see number 13? Can you see the structure of the maize grain?
Yes.
तो 5 आ रहा है समझ में? The numbers 5 समझ में आ रहा है? The parts number 1, 2, 3, 4 वो समझ में आ रहा है? Yes? No? That's not clear? Acha, not a problem. This sometime later I will give you when I come to the class. Now, I'll keep on giving you the questions. You will try to write down the answer. Write down the questions. Number 1. Write down the questions. Write down the question. Number 1. Give 2 examples each of. Give 2 examples each of. Give 2 examples each of. Number 1, you write dicotyledonous, albuminous seeds, dicotyledonous albuminous seeds, dicotyledonous albuminous seeds. Dicotyledonous albuminous seeds, and number 2, dicotyledonous X-albuminous seeds. Dicotyledonous X-albuminous seeds. आंसर लिखो, दोनों का आंसर फाइंड आउट करो. I'll give you 1 minute time. I gave you 2 questions. Find 2 examples each of dicotyledonous albuminous and dicotyledonous X-albuminous. Dicotyledonous albuminous and dicotyledonous X-albuminous. करो. Write 2 examples each. I asked you. लिखो. दो example each for dicotyledonous albuminous, dicotyledonous X-albuminous.
I'm done.
What are the examples?
Some dicotyledonous albuminous seeds are coffee and custard apple.
Very good.
Dicotyledonous albuminous seeds are peach and mango.
Okay, very good. Now, next question. Next question is, next question is again 2 examples each of monocotyledonous albuminous and monocotyledonous X-albuminous. Monocotyledonous albuminous and monocotyledonous X-albuminous. Monocotyledonous albuminous, monocotyledonous X-albuminous. लिखो. Done.
Ma'am.
Next question. What is the function of? What is the function of cotyledons in P? 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 Right, one sentence answer only. One sentence answer only. Over. Done. Now, can you see these questions? Comment upon the following. Comment meaning give reason. You have to give reason. You have to write one reason. The first one, let us do. Ploughing the fields is must for the process of germination of seed. Ploughing the field is must for the process of germination of seed. Right on this question. Ploughing of the field is must for the process of germination of seed. Let us write down the answer. Did you write down this question? Ploughing of the field is a must for the process of germination of seed. Did you write down this question? Likho, answer likho. What do you think you should write down the answer? Ploughing of the field. Why is it necessary for germination of the seed? Ploughing of the field. Why is ploughing of the field so necessary for the growing of the crops? Germination of the seed. The answer is ploughing of the soil. Likho, ploughing of the soil. Ploughing of the soil makes the soil porous. P-O-R-O-U-S. Ploughing of the soil makes the soil porous. Makes the soil porous and creates air spaces. Creates air spaces so that seeds get adequate amount of oxygen. Creates air spaces so that seeds get adequate amount of oxygen. Seeds get adequate amount of oxygen. Done?
Yes, ma'am.
Come to the next question. I'll give you this question. This you will write in your words. I'll give you 1 minute for writing down the answer. Likho, next question. If you are done with the previous, write down the next question. Soaked seeds, soaked, S-O-A-K-E-D, soaked seeds germinate more easily. Soaked seeds germinate more easily than dry seeds. Soaked seeds germinate more easily than dry seeds. What to write for this answer? Think and write down. 1 minute time. Soaked seeds germinate more easily than dry seeds. What should the answer be now?
I've written that the seed is dormant and of water it is active.
So, soaked seeds contains adequate amount of water, which helps in the process of germination. Is not so? That is what you are writing. Soaked seed has adequate amount of water, which helps in the process of germination. Did you all write this?
Yes.
All of you?
Yes,
Done. Come to the next question. Come to the next. Third is most seeds do not, most seeds. Third question. Third question. Seeds of most of the plants. Seeds of most of the plants do not germinate. Seeds of most of the plants do not germinate. Seeds of most of the plants do not germinate even under. Seeds of most of the plants do not germinate even under suitable condition. Even under suitable condition. Think क्या लिखना है? What will you write? Think. I repeat the question. Seeds of most of the plants do not germinate even after or even in presence of suitable condition. Think what to write there.
Because the seed has not undergone physiological maturation.
Because
the seed has not undergone physiological maturation.
It's not clear. The seeds have not undergone what you're saying?
Physiological maturation.
No, not that. Not that. Seeds of most of the plants do not undergo germination even after getting favorable condition most of the time. Your answer. Because?
The seed is damaged.
Arey no. You are supposed to write, this is because most of the seed have a period of dormancy. This is because most of the seed have a period of dormancy. Even after getting, I'm telling you the answer, you will write it in your own words. Most of the seed, even after getting a period of, most of the seed, even after getting favorable condition, even if one of the factor is little bit up and more or less, then also they undergo a period of rest or dormancy. So your answer is, most seed have a period of dormancy. That is the reason, even after favorable conditions, they may not germinate. Likho. Did you understand what I told you? Most seed have a period of dormancy, period of dormancy. Dormancy meaning the seed does not show any physiological function. Most seed have a period of dormancy, hence, even in presence of favorable condition, they do not germinate. Write down. Done? So few questions, this way we are done. Now, write down 2 questions, you will write on your own. Number 1, differentiate between alurone and endosperm. Differentiate between, number 1, differentiate between alurone and endosperm. Alurone and endosperm. And number 2, cotyledon and scutellum. Cotyledon and scutellum. Cotyledon and scutellum. Only one difference to write. Think, I'll give it 1-2 minutes time. 1 minute per question. Think what to write. Scutellum and cotyledon में क्या difference लिख सकते हो? One point. And the other one I said is what? Alurone and endosperm. 1 point. Only 1 point to write. Bhakti, what are you doing? Right. What is the answer, Bhakti? What to write? Number 1. Allurone and endosperm. What is the difference to write? Allurone and endosperm. What is the difference to write? Bhakti, can you hear me?
Yes, I have not written.
Who has written this answer? Shreya, are you done? Allurone and endosperm.
Ma'am, endosperm is rich in starch and erythrol is rich in protein.
Okay, good. That is what you have to write. Allurone is rich in protein, endosperm is rich in starch. Allurone and endosperm both present in a monocot seed. And the next one I said what? Next I
Cotyledon and scutellum.
scutellum. Cotyledon and scutellum. Cotyledon and scutellum. Okay, now give me the answer there. What's her name? I forgot again. Ashita. Ashita bolo. Answer bolo. Scutellum and cotyledon. What answer to write? One difference only. Scutellum and cotyledon. What answer to write? What difference to write? You will, never mind if you have not written. Can you tell me the answer? What to write? Think. Cotyledon और scutellum में कुछ difference है? Cotyledon and scutellum में क्या difference है? हाँ? Say a bit louder, otherwise the microphone is quite far away. Say a bit louder, otherwise I'm not able to hear.
With containing food for the embryo and protect it, and they are in diploid seed and embryo and the scutellum is a single cotyledon in monocot seeds.
Scutellum is the single cotyledon of monocot seed. Scutellum is the single cotyledon of monocot seed, whereas cotyledons are the fleshy part of the dicot seeds. That's the first difference. Any other difference that you have to write? There are many. But agar kuch aur likhna hai, then cotyledons store abundant amount of food. Scutellum does not contain or scutellum is not a rich source of food. Cotyledon store abundant food. Scutellum is not a rich source of food. Scutellum is not a rich source of food. Complete karo. Can you see this number 10?
Yes, ma'am.
Are the questions clear to you? Number 10.
Yes, ma'am.
So karo, solve karo number 10. And number 11 last, we'll complete with this chapter. Number 10 solve karo.
Right, on the screen. What is the aim of the experiment? I'm not getting the second question.
Acha. What is the aim of the experiment? All of you, look into that question there. Study the diagram first, and then the first question. What is the aim of the experiment?
To show that water is required for germination.
Yes. Purpose of beaker B. In beaker B, what is there? Dried seeds. No water. What is the purpose of beaker B? The purpose of beaker B or beaker B acts as a control. That is what you are supposed to write. Beaker B acts as a control. 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 Beaker B acts as a control. What is the meaning of control? It is a setup जिसपे कोई observation नहीं रहेगा even after 10 hours, 12 hours. And that is how you will understand what is the comparison with the other setup जो observation दिखाएगा. Two setups here. A is showing observation, B will not show observation. So such type of setups which we just keep for comparison and which will never give us any observation, usko bolenge control setups. This is a control setup. B is a control setup. Done?
Yes.
Now, next is draw the diagram of beaker A as it would appear after few days. After few days, how did you draw the diagram? How will you draw the diagram?
Ma'am, the seed will germinate of beaker A.
So every seed will have some sprouts coming out.
Yes.
Every seed will have some sprouts coming out. So that is what you have to show. Every seed will have some sprouts coming out. So that is how you should draw the diagram. Would the result be same if instead of, would the result be same if instead of dried peas that have been killed by heating in an killed by heating in an oven are used, would the result be the same if instead of the peas which are killed by heat? So you have to focus on beaker A. So they are saying that would the observation in beaker A would be the same even if heat treated or heat killed peas we use. Put the peas in the oven and then you use it for the experiment. Would the observation be the same in that case?
Yes, ma'am, because the seed will die.
Because?
Seed will die.
So will the result be the same?
Yes, ma'am.
Would the result be the same?
Yes, ma'am.
Would the result be the same? We are talking with reference to beaker A, no? Beaker A is showing sprouting, beaker A is showing germination normally. Now they are saying instead of this common soaked seeds, don't take this soaked seeds. Take seeds which already you have dried up, heated up in the oven and then you use them.
No, ma'am. I thought that A and B will be the same.
No, they're talking with reference to A. See, they draw the diagram of beaker A as it would, and then they are talking about the question, would the result be the same? Would the result be the same? No, your answer is no, the result would not be the same. Your result would, the result would not be the same. No, the result would not be the same as the seeds would be killed. As the seeds would be killed due to heat.
Yes.
(13) Are you able to see now 13?
Yes.
Chalo, last question karo and then we wind up. Last question, number 13, complete karo.
In the box. 13.
Now let us complete with these questions. The first is name the parts label 1. Name the parts label A to F. A is?
Endosperms.
B is?
Scutellum.
C?
Coleoptile.
D?
Pupule.
E?
Radical.
F?
Coleorhiza.
Now, Ashita, what is the function of coleoptile and endosperm? What is the function of coleoptile and endosperm? Endosperm you told also earlier. Coleoptile का क्या लिखना है? What is coleoptile's function? What to write? Coleoptile. See in the figure, what is the coleoptile surrounding? Coleoptile surrounds what? What is it? Coleoptile is surrounding which structure? Ashita, can you hear me?
It surrounds the plumule.
So what is the function that you should write?
It protects the plumule.
Protects the plumule. Protects the plumule. Come to the next. What kind of seed germination does the maize show? Maize hypogeal. What kind of germination does the maize show? That is hypogeal germination always.
Yes.
So see that you are done with all the questions in your book also, in your textbook. The questions, other than that, the basic differences in in your textbook, there are certain basic questions about the differences between hypogeal, epigel, and then the differences between coleorhiza, coleoptile, maize, and the bean sage. So these are some common questions that you should go through.
Yep.
Two examples of viviparous plants that also you should remember. So from this chapter, progress check in your textbook. Did you complete the last progress check?
Yes.
The last progress check, did you complete in a dry seed, the embryo is in? Did you complete that set of questions? अगर नहीं किया है तो वो कर लो. See that all the set of questions you are done in the textbook.
Yes, I'm doing.
From the rest of these chapters, that is, what are the other three chapters which are majorly cut out? Portions are cut out. Which are the other three chapters?
Ma'am, 5 kingdom classification, diseases, aids to health.
Aids to health, any confusion?
1 minute. No, ma'am.
Okay. And diseases also, no? That chapter also.
Yes.
Done?
Yes, ma'am.
So we are done with this chapter also, and for your portion, for your test, all the portions are now covered, right? Any confusion hai? Any query hai? Bhakti, Ashita, anything to clarify?
No, ma'am.
Bhakti,
No, ma'am.
nothing to clarify. So I stop sharing. So from when are your lectures getting cancelled, from tomorrow or next week? Next week? So Bhakti and Ashita, no exam, no now? You also have Bhakti also exam? Ashita's exam when? Ashita, when is your exam? It's over already. Huh? For you all, it's already over.
Yeah.
So it's only for Shreya and Bhakti. I did not understand. For you, exam is there?
From October 8th.
From October 8th. Okay. Chalo, all the best. Okay. Wind up today.
हाँ मैडम जी हो गया.
Yes, yes, yes. Thank you so much.
Yeah, ma'am. Thank you. क्या बोल रही कोई नहीं तू भी पूरा अपनी मम्मी को कॉल कर